discussion on drug with legends, with association of Department of Dravya Gun and Agar Tantra. On the behalf of department, I would like to welcome our respected Vice Chancellor Sir, Professor Sanjeev Sharma Sir, Pro Vice Chancellor Ma'am, Professor Ram Murthy Sir, all the Dean of National Institute of Ayurveda, all the HODs of the department, and all PG, PhD scholars of Department of Rasastra and Bhaishajya Kalpana, Department of Dravya Gun and Agar Tantra. And I also welcome our guest speaker, respected Professor S.S. Savrikar Sir. Give a big hand to Professor Savrikar Sir. And I also welcome all the participants who have joined at different places in India. Let's begin the program with the Dhanvantari Vandana. Thank you, sir. Now I would like to invite HOD Sir, Professor Anupam Srivastav Sir, for welcome address, brief introduction about our webinar.
கன்னியாகுமரி மாவட்டம் தேங்காய் மீன் துறைமுகத்தில் கடலில் மாயமான மீனவரின் உடல் மீட்கப்பட்டுள்ளது. உடலை வைத்து மீனவர்கள் போராட்டம்.
Dr. Ritesh,
switch off your audio system. Please switch off. I again request you. Please switch off your audio system. Otherwise, the purpose gets defeated.
please सबको म्यूट होने को भी बोलिए, डिस्टर्बेंस आ रहा है.
Sir, you are not audible. So please unmute yourself.
कुछ सुनाई नहीं दे रहा है कुछ आई डोंट नो
I am audible, expert, sir.
Yes, sir. Yes,
हाँ,
sir. You are
अब सुनाई
audible.
दे रहा है actually.
yes okay
Thank you once again. Welcome to all who has joined through virtual platform and physically on this starting program of the drug discovery, drug knowledge, drug understanding, what you call. First of all, on behalf of entire team of NIA and also from the Rashas, Dravaguna and also from the Agattan, an entire team of the RAV, I welcome, I extend my Kritagyata Gyapan to Vice Chancellor Sir, NIA, Pro Vice Chancellor Madam, and all today's experts, Savarkar Sir, my guide, Professor C.V. Jha Sir, who has joined us virtually, industry partners, C.V. Katiyar Sir, and whom I am not able to name them. I welcome you once again all. And for my base of the heart, I also extend my welcome. I welcome to the senior officials of the Ministry of Ayush, our Honorable Minister Sir, both of the Ministers Sirs, Secretary Sir, Special Secretary Sir, and Advisor Sir, for mentoring, for guiding, for promoting us, and for encouraging us for the development of the system, for development of the knowledge, for sharing of the knowledge. By their mentorship, by their guidance, it will not possible to propel any education system in the country, especially of the country also, especially for the Ayurveda. Very quickly, we all understand the importance of the knowledge, importance of the drug in current scenario. And this, we all understand the quest for the knowledge, the quest for the drug, the quest for the longevity, quest for the well-being has been always a part of human interest since existence of the mankind. And similarly, the philosophy goes more adding on when it comes to drug. The knowledge of the drug has been since the mankind. And if you turn the pages of the Vedas, it clearly speaks about Purva Jata Sangya and also the origin of the drugs from the Brihaspati. This is all about the drug information. Very quickly, I want to share. And there has been always quest. There has been always quest for the mankind. If you talk particularly in terms of the Indian context, this goes always for the reaching the level of the salvation. Definitely, our text says this salvation could not be reached until and unless the use of the dramatic medicine which we are having, like Ras and Rasayan. It has been rightly said in the text, Pind pate chayo moksha, sacho moksha nirathaka, Pinde to patite devi gadhopi vimochet, tasmat sanrachet pindam rasayas rasayani. Pind means the body cannot be procured, and you can't reap the salvation until and unless you use the Rash Aushadhis, Ras and Rasayan Aushadhis, that the salvation could be, highest degree of the reach could be available. This, we all understand in a current scenario, there are a huge hue and cry about particularly Ayurveda drugs, and the interest is compounding the system. We all understand discovery of a new drug is $1 million affair. It is $1 million affair, and in a case, if the importance compounds when we talk about particularly the development of a new drug, and when the population, more than 80% of the population of the even world is using somehow traditional or their own system of the medicine other than those synthetic or Western medicine. So this way the importance is higher and the discovery is also tough. Similarly, if you see the database, the knowledge database which is available at our end, particularly at Ayush and or Ayurveda end, this we have a huge database of the knowledge in terms of the patient, in terms of the treatment methodology, in terms of the patient care, in also terms of the pharmacology. This could be well translated if the proper suitable drug is used, and if will pave the path for the new concept of the designer medicine. हम सारे सुनते हैं कि designer medicine के बारे में, हमारे पास बहुत सारी information है which could be translated, और हम we all understand there has been disastrous effort in past में क्या effort हुआ? Destruction, deliberated disastrous destruction of the Indian knowledge. वो terms of चाहे ब्रिटिशर्स रहा, हमारे knowledge को कमतर आका गया, हमारे knowledge को हमारे एक मैकाले ने एक टीचिंग सिस्टम लाया जिससे कि एजुकेशन सिस्टम हमारा इस तरह कहा गया कि नहीं, जो मॉडर्न सो कॉल्ड वेस्टर्न सिस्टम है, दे आर द मोर बेटर एंड देयर आर मोर इट कुड बी यूज्ड, बट डेफिनेटली वेस्टर्न सिस्टम का नॉलेज होना चाहिए, वेस्टर्न मेडिसिन भी आप जाने, लेकिन वेस्टर्न सिस्टम और जो हमारा अपना आयुष सिस्टम है उसको मैं इस तरह लेमन लैंग्वेज में कहता हूँ, एक हमारी मौसी है, दूसरी हमारी माँ है. वेस्टर्न मेडिसिन कुड बी आवर मौसी. मेरी मौसी कितनी भी खूबसूरत हो लेकिन मेरी माँ जैसी नहीं हो सकती. ये कंसेप्ट आप जरूर रखें. ऐसा नहीं है कि आज हम ग्लोबल विलेज में रहते हैं तो वी मस्ट अंडरस्टैंड द अदर सिस्टम्स आल्सो, बट वी शुड ट्रस्ट ऑन व्हाट द सिस्टम वी हैव. वी मस्ट ट्रस्ट अपॉन व्हाट वी hear since the ages. So this is all future. We understand for the medicine, future is for the more holistic, personal, intervention based, complete use based medicine, which could be further maximize the benefit of the humanity, benefit of the common man. With this small and this platform, I am hopeful with the blessing of all the seniors, with the blessing of our own VC sir and all the seniors, we will definitely reach. We will in this Aushad Jigyasa discussion, this series which we are starting. This is basically a kind of the information sharing series, a kind of the discussion platform, a kind of the knowledge sharing. It could be a kind of a tutorial classes. I am thankful to my other colleagues, other heads of the concerned department, Dravaguna and Nagattan. I want to clarify this platform. This is not limited to only these three departments or these specialties. The floor is open. The floor will be open for the discussion of the drug right from its origin up to the maximum use. Ultimate aim is the cure the ailing humanity, how the drugs could be beneficial, what could be their different parameters, right from the quality parameter, right from the effectiveness, right from the safety. All will be discussed on this platform, and we are fortunate enough that Professor S.S. Savekar, he is a legend. He is a legend in the Shastra. He would like to share his experiences. He would like to propel in inaugural series how we should take the further, how we should take this platform further. It should be beneficial. With this background, I have taken a lot of time. I'm sorry for that. With this background, I again share stage to my colleague Dr. Ritesh to invite the further seniors. Over to you, Dr. Ritesh. Thank you.
Thank you so much, sir, for brief introduction about our program. Now, I would like to invite our Pro Vice Chancellor, Ma'am, Professor Meeta Kotecha, Ma'am, for few words. Professor Meeta Kotecha.
नमस्कार. डीसी सर, प्रोफेसर संजीव शर्मा सर के गेस्ट, प्रोफेसर सावरीकर सर, प्रोफेसर अनुपम जी, यहाँ के हुए सभी जोड़ी टीम और बाहर के जुड़े हुए एनआईटी के बाहर के जुड़े जितने भी और ये औषधि जिज्ञासा शुरू करने के पीछे का कारण तो अनुपम जी ने बता ही दिया. इसमें जैसे उन्होंने बताया कि ये 3 डिपार्टमेंट ने एक आइडिया से शुरू किया है, पर उसका मतलब कतई नहीं है कि ये 3 डिपार्टमेंट इसमें पूरी जानकारी जब तक पूरी नहीं हो तब तक से शुरू करके द्रव्य का एस्पेक्ट, शास्त्र का चिकित्सा का एस्पेक्ट, काय चिकित्सा वाले कर्म का उपयोग कर रहा है, कर्म वाले बताएंगे. एक द्रव्य को लेकर उसका पूरे सब एस्पेक्ट्स में जानना, ये जिज्ञासा का उद्देश्य है और सोच के ये शुरू किया जा रहा है. और आज सब लोग यहाँ हुए हैं कि बहुत अच्छे से चले। इसमें सबका पार्टिसिपेशन होना चाहिए, ये ऐसा नहीं होना चाहिए। जितने भी स्कॉलर जुड़े हैं, आप जो भी आप सकते हो और रहेगा तो बहुत अच्छा होगा। जिस उद्देश्य से हम क्या है, उस उद्देश्य को पूरा करेगा। मेरी बहुत बहुत शुभकामना है कि अच्छे से चलेंगे और इससे आयुर्वेद के लोग हैं उनकी शक्ति बुद्धि होगी और उनको कहीं नई चीज जानने को मिलेगी, ज्ञान में अभिवृद्धि होगी। बहुत बहुत धन्यवाद और बहुत बहुत शुभकामनाएं। नमस्कार।
For blessing words. Definitely we will include clinical departments also in future. Thank you so much, ma'am. Now I would like to invite respected VC sir, Professor Sanjeev Sharma sir, for
thank you, Ritesh ji, and very good afternoon to everyone. Am I audible? Am I audible?
Yes, sir.
Yes, sir.
Thank you. Okay, thank you. So, most respected, the legend of Ayurveda and Rashastra, pioneer, our mentor, our guide, Professor Savrikar ji, all other esteemed and very senior legends of Rashastra and Ayurveda, like Professor Jhaji, Katyar ji, Professor Meeta Kotecha ji, Professor Anupam ji, all fellow colleagues of the faculty of Rashastra, all dear participants off the line and online. First of all, my heartiest greetings on the very pious occasion of Ganesh Chaturthi to everyone, and may Lord Ganesha provide us all kind of happiness, prosperity, and all what we desire. So today, this is a very, I can say, auspicious occasion that we are going to inaugurate. And this is inaugural session of a very important series of the lectures, that is Oshad Jigyasa, discussion on drug with legends. This is an initiative, in fact, has been taken by the Rashastra Department of National Institute of Ayurveda and brainchild of Professor Anupam ji. And definitely this, what they have, the idea they have conceived, that will be well taken by all the aspirants of Ayurveda, because topic is very interesting. Aushad. As we know that when we talk about the profession, medical profession, then patient is in fact the fulcrum of our whole of the medical profession. Maybe be in the academics or research or in clinical practice or in industry or in other areas of medical field. But the patient is a fulcrum and we all revolve around the patient. Because every effort which what we are putting there is for the welfare of the patient. Or sometimes it is also for the promotion of the health. So both the things go hand by hand. If it is for the treatment purposes, then the patient is in the center, and if it is for promotive purposes, then the person is in the center. But when we talk about treatment particularly, or for the rejuvenation or promotion of the health, then there are so many systems. It may be drug therapy, it may be non-drug therapy or non-drug interventions, but the mainly for the world, maybe any system of medicine, the drug is very crucial and very important, and we should know about the drug. In Ayurveda, if we divide the whole period till now, we can divide the period into three parts: Vedic era, Samhita era, and the present era, post-Samhita era. In Vedic era, although the use of the drug was very common during those periods also, but the use of Daivapya Ashray Chikitsa, that was also there. During Samhita period, the use of drugs use of medicine that increased, and after that in post Samhita period, there was revolutionary changes in the Ayurvedic system of medicine, and during the 8th century, I think, the Rashid Nagarjun, he revolutionized the whole scenario of Ayurveda. And then Rasashastra, which popularly today we know, that came into existence and as a branch of, and it was merged with Ayurveda, and it became into the practice and into picture also. So now, Aushad, it is a very diversified topic. Aushad, it may be from plant origin, it may be from animal origin, or it may be from mineral origins. But when we are in practice, or when we are in research, or in academia, or in industry, or even if we are the farmers or growers of the medicines, or we are mining the medicines from somewhere, then definitely we should have some basic knowledge of medicine, or drug or medicine. And it is this discussion on these medicines is very important for everyone who is involved directly or indirectly in the this medical field. And for particularly students and clinical practitioners and researchers, the whole cycle of drug he should know. That if it is from animal origin, plant origin, then how and where it is produced, how it is produced, how it grows, and how it is collected, how it is stored, and then how it is processed, and how it is then manufactured, and then how it is given to the patient, in what form, what dosage form, what dose is given, and then what are its indications, contraindications, and then what are its side effects, and what are its other aspects of, means whole cycle of drug, it is very important. Similarly, for mineral origin drugs, same cycle one should know. And I hope that this series of discussions, which will go on continuously, every aspect of the drug will be discussed, and I think all the kinds of experts, they will be involved in during all these sessions, and we all will be very much beneficiary out of these discussions, that because the knowledge which will be shared by our legends, our experts from different spheres of the expertise, maybe from Rasashastra, maybe from Agatantra, maybe from Dravya Guna, or from some clinical departments, or from modern medicines, or from pharmacologists. Whosoever is the expert, definitely he will be sharing very important his ideas and inputs, and he will be giving, and that will be beneficial mutually for all of us, not only for the student fraternity, but for the teachers, for the clinical practitioners, and for even for the industry. These discussions will be much beneficial, and I definitely hope and wish all the best for this series, which a very daring decision definitely Department of Rasashastra has taken, and I really wish that this should be a most successful series of the lectures, and it will be most beneficial for the students. So my best wishes are there, and every kind of support from the institution side I shall provide for the success of this event, this, I can say, the series of events. Every kind of support will be given from my institution side. My heartiest welcome on behalf of my institute and Ministry of Finance to all the legends, all the participants who have joined offline or online on this platform. Again, my best wishes, and thank you for giving me this great and wonderful opportunity to be here with you people to share my views. Thank you very much.
Thank you so much, sir, for your blessing words. definitely we are revolving around the patients, and we are for patients it is very important to discussion on medicine, because for the promotion of health or any disease condition, medicine is very important substance. So this is very important thing. Now I would like to invite respected Professor Sanjeev Sharma sir, Vice Chancellor sir, to inaugurate our international conference brochure.
On behalf of entire NIA, I will request Vice Chancellor sir to kindly release first brochure, first round of information for the coming event which is planned. Over to you, sir.
Thank you. Really, this is again a very wonderful opportunity for me to announce and to reveal the first this brochure, or first circular of the proposed international conference on Ayurvedic pharmaceutical sciences, which is proposed to be held in NIA campus on 24th and 25th February 2023. Really, this will be a big event, wonderful event for everyone, and I request everyone who have joined or who may not have joined, please circulate this among all persons, all the stakeholders, so that everybody can get an opportunity and benefit out of this. So my best wishes again for the success of that conference, which is going to be held on 24th and 25th February 2023. That is International Conference on Ayurvedic Pharmaceutical Sciences. Thank you very much, and my best wishes.
Thank you so much, sir. Your kind blessings always with us. Now we will start our scientific session, and before starting our scientific session, I would like to share some brief bio sketch of our respected Professor S.S. Savrikar sir. Professor Savrikar sir is MD, PhD, and former professor and head of the department of Rasastra Bhesajya Kalpana, R.A. Poddar Medical College, Mumbai. Sir have vast experience. Sir had advisor in Government of India, and sir was Vice Chancellor of Gujarat Ayurved University, Jamnagar, and sir was Dean, Faculty of Ayurved, MUHS, Nasik. Sir having teaching experience of about 40 years, and sir is chairman of scientific body in Pharmacopoeia Commission of India. Sir have nominated as a member of organizing body CCRAS, and member of BHU Court Varanasi, and member of Management Council MUHS Nasik. Sir have got lots of awards. In them, one of them, Nagin Das Saha Award 2019. Sir have so many publications, and one of them Ras Chandanshu Hindi commentary, and much more. Now I would like to invite respected Professor S.S. Savrikar sir for today's lecture.
Over to you, sir. Over to today's speaker, Savrikar sir.
Yeah, thank you very much. Let me share my laptop. Can you give me sharing? Can I share it now? Excuse me. Can I share?
Yes, sir.
Okay. Okay. I think you are not given me sharing. Could you be sharing because it is not being shared? Excuse me, organizers.
Yes, we are managing.
Couple of minutes, sir. Please give a couple of minutes. We are just
Okay.
some glitches are.
Okay.
Are you able to share, sir? We have submitted. Inspector, sir?
Yeah, just wait. I'm trying to do it, but no, still not. Just wait. From here also I'm not able. Windows. No. शेयर नहीं हो पा रहा है स्क्रीन, जरा देखिए क्या हो रहा है. शेयरिंग आपको देना पड़ेगा, शेयरिंग राइट से निकलते हैं.
Sure, sir. Give a second, sir, please. अपनी शेयरिंग स्टॉप कर दीजिए.
Yeah. Can I see my screen now? Can you see my screen?
No, sir, no.
No.
Yes, coming up. Yes, it is there.
Okay, thank you. Thank you very much. Okay, let me start my lecture, please. Honorable Dynamic Vice Chancellor Professor Sundar Sharma ji, Co-Vice Chancellor Madam, Dr. Srivastava ji, Head of Department of Shastra, Head of Department of Agat Tantra, Dravya Gun, and the experts from all over India, particularly Dr. Katiyar, my friend, Jha sir, and other experts who have joined this webinar online, and all the teachers and students from NIA and all those who have joined this particular webinar. Thank you very much for giving me an opportunity to give an inaugural address to this very important topic regarding knowing about the Ayurvedic drugs. I'm asked to give an overview of Ayurvedic drug. But when I started thinking over it, when 2-3 days back, I got a call from Dr. Srivastava that, sir, I request you to deliver the first lecture on this particular series. Then I started thinking about it. And then I thought it's suitable to give it an overview. But how, whatever is missing, that I thought I should give some thread should be there to deliver my lecture. So over the time which I'm looking after, so many things are happening in drug industry, in academics and practitioners. People are using Ayurvedic drugs and using them by different methods. People are using them as just like a modern medicine. Some people are using like an Ayurveda, like that. Even in research also, there is no definite strategy behind the research also regarding the, as far as drugs are concerned. I find that there is some missing link between the principles and practice of Ayurvedic drugs. And unless we find out that missing link, nothing much important will occur. We will get some very minuscule things about which we will be happy. Some people will say, no, we have found out this particular thing. We have found some hypoglycemic, we have found some adaptative drug. People will say like that. Industry will be happy because their sale will be increasing much more. Recently, recently I heard something that the industry sale of Ayurvedic drugs has been increased within these 2 years, about 10 times or like that it has increased. So industry is very happy. They are least worried about what exactly what their drugs are doing. But as an academician, as a practitioner, are we happy about whatever is happening? At least I am not happy whatever is happening regarding as far as Ayurvedic drugs is concerned. I am happy because Ayurveda is being propagated everywhere. People are talking about Ayurveda. It is going abroad. People are going abroad, getting many places. Ministry is established. Many new things are happening. People are accepting Ayurveda. Okay. But what about the real things? Whatever are happening as an academician, as a practitioner, am I happy? I am not happy, my friends. I'm sorry to say that. People will be injured because of my saying this thing. But it's what? I find there's some missing link between principles and practice of Ayurveda. So I'm trying to see what that link is. It's the link which I'm saying is missing is according to me, I'm saying that. It may be not be right for other people. But I think this is what is missing in our practice of Ayurveda. So to find out this link, first, as in the initial introductory speeches, Dr. Srivastava as well as the Honorable Vice Chancellor, my friend Dr. Sharma ji also said, that a drug is a very important tool in the hands of every practitioner. We say, Bishak Dravyani Upasthata. Now, Bishak Dravyani Upasthata means Bishak comes first in Pachaturka, and the Aushad Dravya comes second. That means if a Vaidya, a physician, is the most able, the first tool in his hand is Dravya. May it be a medicine, may it be a food material, whatever it is. But that is a tool with which he is going to treat the health or manage the health or manage the disease condition, whatever it is. So let us understand what exactly you mean by Aushad or Dravya, because the theme of this particular webinar is Aushad Jigyasa. So first Jigyasa is what is Aushad? What is exactly the drug which we are talking about as far as Ayurveda drug is concerned? Of course, this definition of the Bheshaj or Aushad is applicable to everything what is known as a drug. But what Ayurveda thinks about that particular thing, this is very important for us to understand. So for that to understand, first basic principle is that for everybody, that you have to apply the correct principle to understand these things. Unless you apply the correct principle, you won't understand anything. That's very important. So that's why you know that there are Siddhant, there are 4 types of Siddhant. Sarvatantra, Pratitantra, Adhikarana, Abhyavagam. We are not today discussing the Adhikarana and Abhyavagam. This is not concerned with us. But today 2 Siddhanta principles are most important. Sarvatantra Siddhanta and Pratitantra Siddhanta. So these 2 principles are important. Why important? Because Sarvatantra Siddhanta is general principles. As though Sarvatantra Siddhanto Nama Tasmin Sarvansmin Tantre Tat Prasiddha. That means this particular principle is applicable everywhere as far as that particular subject is concerned. For example, if it is a medicine, as far as our medicine, whatever medicine is there, whether it is an Ayurvedic medicine, whether it is a homeopathic medicine, whether it is a modern medicine, whatever medicine is the basic subject. So in all medical sciences, the principle will be applicable. Whatever it is. Let us take one principle, Sarvada Sarvabhavanam Samanyam Vriddhikaranam. This principle is applicable irrespective of any particular medical science. It is applicable to modern medicine, it is Ayurveda, it is applicable to homeopathy, etc., etc., etc. Whatever it is. So this general principle is okay. But there is another type of principle, which is Pratitantra Siddhanta, subject-specific principle. Within that system also, there are different systems. So the principle which is applicable, as far as applicable means actually principle can be applicable everywhere. But to understand a particular statement in that particular system of medicine, you have to understand that particular principle. That principle will be applicable only for that particular system, not for other system of medicine. So that's what it is a subject-specific principles. So Pratitantra Siddhato Nam Tasmin na Ekaikam Smin Tantre Tattat Prasiddham. Very important. So to understand Ayurveda as Ayurveda, we have to understand this Pratitantra Siddhanta. What is the basic principle applicable to Ayurveda? So what I think is missing link is application of specific principles to the practice Ayurveda. That is what is missing. What we are doing, we are looking to Ayurveda by applying generalized principles. The principles which are applicable everywhere, you are applying here also. But to understand some particular subject, very specific subject, we have to apply the specific principles. Unless you apply the specific principles, you can't practice. You can't practice that system. You won't understand that particular system also, that particular subject at least. So the most important missing link is for application of specific principles to practice Ayurvedic chakra. Everywhere we are missing that link. We are forgetting the specific principles. Whenever you are practicing Ayurveda, you are doing research in Ayurveda, you are teaching Ayurveda everywhere, you are missing this specific principle. You are trying to apply generalized principles whenever you are teaching Ayurveda. So this is the missing link which I think, and now I am trying to see what that particular missing link is. So that's why the basic principle behind this particular thing is understanding applicability of Ayurveda specific principles is most important. Because unless we understand those principles, we are forgetting those principles. A very pitiable condition actually. It is generally said that when you are in Rome, do as Romans do. It's a very general proverb it is. So it is applicable. So when you are in Ayurveda, do as Ayurveda system is doing. What Ayurvedic seers were thinking, what Ayurvedic scientists were thinking. When you are reading Charak Samhita, you have to understand the principles of Charak Samhita. You can't apply the principles of some Goodman Gilman to understand the principles of Dravya Guna in Charak Samhita. You can't apply those things. Maybe superficially you may be able to understand the basics of to reach the minutest part of that subject. You can't apply the Goodman Gilman principles or modern pharmacology principles to Ayurveda, my friends. At least unless you understand the correlation between the basic principles and the pharmacological principles. Without doing these things, we are just doing haywardly, we are applying the modern medicine pharmacological principles or other principles of medicine to Ayurveda. And that is why we are failing miserably. We are failing. Maybe we are very successful in increasing the sale of Ayurvedic drugs to 10 times or 100 times. You will be richer, no issue. But will you be richer by knowledge? You will be richer. It is jokingly said that Saraswati and Lakshmi do not go together. Are we going to prove that principle that Saraswati is Ayurvedic Saraswati is far long, at a far distance from the Ayurvedic Lakshmi? I think as a true academician, true practitioner, you don't want this to happen. You want to go Lakshmi and Saraswati, Ayurvedic Lakshmi and Saraswati hand in hand together. If that is what is important, you want, then you have to understand, my friend, the basics. So to understand the basic principle, if I am not audible, please let me know, because I am continuing. Am I audible? Please,
yes, sir. You are audible, sir.
And I have 1 full hour now, because already more than half hour has been spoiled. I'm sorry, not spoiled. I'd say it was important, but I have no. Okay. Yeah. So the sequence is important. To understand this principle, you have to first understand how this Ayurveda is placed. How it is placed. So I will come back to Charak, because Charak is I will say this, mother of everything, every Ayurveda. You get everything from Charak. This is a source of knowledge, of Ayurveda knowledge. At least I will say the available source, I will say. Maybe initial days, other sessions might have been available today. You see the Sutra Sthan, because Sutra Sthan is important. Even the placement of the sthanas is also very important. The first one is Sutra Sthan. In the Sutra Sthan also you can see the sequence. Bheshaj Chatushka first, Swastha Chatushka, Nirdesh Chatushka, Kalpana Chatushka, Roga Chatushka, Yojana Chatushka, and Anapan Chatushka. And then the last 2 chapters. So this sequence is very important. Usually we don't pay much attention to it, but actually when you read the Samhita, you say to start with every chapter, it has been explained why that particular chapter is placed after the previous chapter. Particularly Chakrabani is doing this thing. To start his commentary, he first says why this chapter is coming after the first chapter. Even the sequence of the shlokas, also sequence of the dravya, sequence of the disease, everywhere there is a definitive sequence is there. It's not that haphazardly it is placed. No. Very important. So this sequential placing of chapters, sequential placing of the subject, you have to understand. If you don't understand it, you will be missing many things, my friends. Many things you will be missing. So there is a different link between these chapters. Bheshaj Chatushka gives you the drugs. Swasthya Chatushka gives the principles of keeping yourself healthy. Nirdesh Chatushka gives the people who will be doing, the factors which are responsible to do all these things, give the maintain the health as well as to treat you, the patient. Kalpana Chatushka, whatever the kalpanas are there which you are using. Roga Chatushka is the different classics of the diseases. Yojana Chatushka is the planning what you are doing, or the therapies. And Anna Panchadashika is your diet, food. So there is a different link. You are doing one thing after another thing after. It's not that you are first doing the yojana and then you are applying the kalpana. No, that's not that. So this linkage is very important. Sequence is important. Try to understand this sequence to understand what is drug, my friends. Very important. Now, as far as today's lecture is concerned, we are not concerned about the other chapter, other chatushkas. We are concerned with the Bheshaj Chatushka. Very interesting, my friend. The first one is the Bheshaj Chatushka Samhita. Main principle is Swasthya Swasthya Rakshanam Aturistha Vikara Prashamanam. So with that way, the Charak has started his deliberations. He has divided this chapter in 4 chapters. Only the first chapter is related to Swasthya Rakshan. To maintain, to secure, protect your health. The first chapter is the Swasthya Rakshan chapter. Whereas the other 3 chapters are Vikar Prashaman, that too they have divided. Antar Parimarjan, Bahi Parimarjan, Ubhay Parimarjan. You can see the sequence, my friend, very important. Apamargatanduli here, related to Antar Parimarjan. Whereas Aragbadhi Adhya related to Bahi Parimarjan. Whereas Shadvadi Chanstashati related to Ubhay Parimarjan. So Vikar Prashaman is by 2 ways. Dividame Bhesham Antar Parimarjan, Bahi Parimarjan. What the drug is doing, very important, my friends. It is either it is expurgating, internal expurgation is there, or the external applications are also there. So the drug, whatever drug you are choosing, it has to either it will be working as an antaparimarjan drug, or it will be working as a bahiparimarjan drug, or some drugs may be having both type of activities. So try to understand these activities and the sequence how it is been placed. Now let us see how they work. So, understanding the work of the direction of the drug. Now, let us come. What is Bheshaj actually? Very important answer. Unless you understand what is Bheshaj, because we are missing this particular thing whenever you are using the drug or preparing the drug or doing research in them. We are missing this particular thing. Bheshaj is defined very nicely. It is defined by Charak. You can see here, it has multiple synonyms. Chikitsitam, Vyadhiharam, Patham, Sadhanam, Aushadam, Prayashittam, Prashamanam, Prakritistapanam, Hitam, Vidya, Bheshaj-namani. There are all our Bheshaj-nama. Now these Bheshaj-nama are very specific, my friends. They have been given with a specific purpose. Try to understand that purpose. You will be wondering why Prayashitta is drug is a Prayashitta. Bheshaj is a Prayashitta. Is it a Prayashitta? You know the meaning of Prayashitta. The basic requirement for a material to act as a drug is, see, evam bhuta bheshajanga rupataya. Sthabar as well as jangam. Both type of drugs, materials. When they will work as a drug, they should have bheshajanga rupa. They should take the bheshajanga rupa. They should have bheshajangatva. That means they bheshajasya bheshajatvam evantaram vyavaharakatam dneyam. They should have Bheshajatva, drugness. They should have the drug potency. They should have a potency to act as a drug. Unless they have a potency, it will not act as a drug. So this to have a potency is the basic requirement of every material to act on. Even though we say, that means we always say that every material found in the universe can be used as a drug, provided yukti purvak you have to use it. Okay, everything is that means yukti is dependent on the bheshajangatva. You have to apply your yukti accordingly. Now, why it is called prayashitta? Very important. Prayashittam is a bheshaja saudhya. Understand? Prayashitta is bheshaja saudhya. Why? Prayashittavat bheshajasya adharma karya vyadhi haratvena. Why vyadhi is happening? Because of the adharma karya. Very important. Padnyaprath, adharma karya. You are doing something, you are doing atiyoga, ayoga, mithya yoga, whatever you are doing, and that is why because you are getting disease. And that disease has to be removed. That's why you are taking a prayashitta by taking the drug. That is why it is called as a prayashitta. Taking a medicine, a prayashitta, adharma karya hara. What is the adharma karya? Adharma karya is a disease, and that has been removed because of the drug. Now, this bheshaj we understood. Now, bheshajam cha dvividam cha tat. Now, see, the interesting part of Charak is that the information is scattered all over Charak Samhita, all over in all chapters. Unless you read all full Charak Samhita, you will not be able to correlate the things. If you read PCF, if you try to read, you won't understand it. We have a habit to take out one particular verb, take out 2-3 shlokas and try and give them that, interpret them. We are missing many things because of that. I find very funny things in that Charak online, which I'm sorry to say this, what has been written. Without having the reference, backward reference, forward reference, it has been translated and we are publishing it universally online. Horrible. My friends, this is very important. Bhesham cha dvividam sastasya. Bheshasya urjaskaram kinchit artasya rogut. Basically, there are 2 types of drugs. Swasthasya urjaskara and artasya rogut. Again, because our word is swasthahitakar and arthanashak. 2 basic classifications. We have seen that swasthasya swasthakshana artasya vikarpashana. Just like that, even the drug is classified into 2: swasthasya urjaskar and kinchit artasya. Urjaskar drug swasthasya is also again classified into 2: vrisham and rasayanam. Dvitiyam artharoga haram. Remember very well. Even the rasayana are artharoga hara. So I will come to that. How do they work? Artasya noganati visheshena jvaradina artasya jvaradiharam. Remember very well. Whenever you talk about artasya roghar, because we are more concentrating on artasya roghar nowadays. Few people try to see the rasayana also without understanding what exactly rasayana is. But most of the people, we try to find out what is the artasya rogharatva in that particular drug. We are more interested in that. Industry is more interested in finding out hypoglycemic drug, adaptogenic drug, immunity enhancers. And they are trying to increase the innate immunity by giving drug. I don't understand that means they have not understood what is innate immunity. Innate immunity cannot be increased by giving some drug. This is basic principles. That means, as I said, do as Romans do when you are in Rome. That we are not. We are entering in the Ayurveda by applying principles of modern medicine, or either way also. My teacher, Gurudev Vyasji, used to tell me one very example, that you have a bed, 6 feet bed is there, and you have a patient whose height is 7 feet. Now he used to ask this, what will you do? That patient should sleep on that bed, which is 6 feet, but his height is 7 feet. What will you do? Are you going to cut the legs of the patient, or are you increase the height of the bed, length of the bed? We are trying to cut the legs of the patient, my friends. We are not looking at the bed size. That is what you are doing. We are all doing. We are fooling ourselves and the world also by doing this thing. Don't do that. So that's all about what is it? The another thing is jvaraji haratvam. Now, yad vyadhi matra haram na tad rasayana. What is the difference? Even rasayana are artasya rognut. Rasayana can also be used as an anti-disease drug. But the difference between only anti-disease drug and the rasayana anti-disease drug, there is a difference. Yad vyadhi matra haram, which is only anti-disease, it is not rasayan. Na tad rasayanam. Yad vyadhi matra haram, na tad rasayanam. Kintu sharira sanyoga dadha dirghayu kartatva sadharana dharma yoga upacharita vyadhi haram rasayanam yuchyate. Now, how the rasayan is doing the vyadhi haratva? How? By sharira sanyog dardhyadi. Sharira sanyog dardhya. Dirghayu kartatva. Like that, upacharita, this thing is doing, and that by that method it is practiced, it is working as a rasayan drug. Now, rasayana also, now the definition of rasayana is important. We are just misleading everything we are listening by doing straightforward classification, translations. We are cheating ourselves and the world also. I don't know who the credit should go to whom. We always translate rasayana as a rejuvenating thing. My friend, rejuvenation is an effect. You have to understand how it acts. The real definition of rasayan is labhopayo hi shastanam rasadinam rasayanam. We are forgetting that. Katham etat rasayanam kriyat. Chakrapani defines, explains it. Katham etat rasayanam kriyat. Labh ityadi. He is not saying rejuvenation. He is not talking about rejuvenation. He says labhopayo hi rastanam rasadinam. Shastanam rasadinam. Rasadinam dhatunam. Shastanam is healthy tissue. My friends, the measures which are beneficial to generate healthy tissue, they are all rasayan. That means healthy tissue generation is rasayan, not rejuvenation. Rejuvenation may be one of the effect, but the most of how does rasayan acts? And it will take time, hours together to explain me how the rasayan act. But in short, the effect I'm saying the most important action is labhopay, labhadayak upay. Labh you can understand, beneficial. Upay also in the measures, beneficial measures for shastanam rasadinam. Rasadi dhatu, shasta dhatu. That means healthy tissue. The material or even the not only drugs but even other measures also which help us, which are beneficial to generate the healthy tissue are known as the rasayan. So yad vyadhi nirghatakaram vakshate tat chikitsate. Now the simple vyadhi matra dravya are not rasayanas. That's why they yat vyadhi nirghatakaram vakshate tat chikitsate. Drug says that I am not describing those drugs in rasayana adhyay, I am describing them in the vyadhi matram chikitsastan. Chikitsartham etavan vikaram yadaushadam rasayan idishcha agre vajikram. In rasayan only rasayan, vrishya and vajikram dravya, those we are describing. Okay, some of the rasayan drugs, they are also acting as an anti-disease, but the principal anti-disease acting drugs are not describing in rasayan adhyay. I am describing on the related chikitsa adhyay. So that is what's important. So try to understand this. Now let me come back to what kashaya or the Bheshaj is. Very important today's topic. Now to understand the kashaya word, very important, my friends. Now kashaya shabdena madhuradinam lavanavarjanam rasayanam. My friends, usually we forget many basic principles. We even don't read also. I don't know how many of us have read the 4th chapter of Sutrasthan, Charak Sutrasthan, Sharverichastha. In the 4th shloka, they have given the kashaya yoni have been given. Kashaya yoni. Because they say Lavana Varja, all the other 5 rasas, they work as a kashaya. Why? Because kashaya tvena paribhashita nam ashaya tvena aushad dravyam uchyate. Which is aushad dravya? Which is bheshaj dravya? Bheshajatva we are discussing in the last few slides. Again, I will discuss that here also. But here I am discussing kashaya. Kashayatvana paribhashitanam ashayatvena aushaddravyam uchyate. That aushadravya should have kashayatva also, because it should have a potential to get processed, because we are processing it. No, it should be able to get processed and produce a drug. That means it should have a bheshajatva and it should have a kashayatva. There are 2 things. Remember well. Now what is kashaya? Kashaya saunayam bheshajatva vyapriyamaneshu raseshu acharayusha. Vyapriyamaneshu very important. Vyapriyamaneshu is spread everywhere. The kashaya term, it encompasses all substances having a potential to function as a drug and produce a drug form. That means that drug form, that material should have the kashayatva and bheshajatva. If any material is not having bheshajatva and kashayatva, you can't produce a drug formulation from that particular material. So are we seeing that bheshajatva or kashayatva in that? That's one. So what is bheshajatva? Bheshajatva is potential to produce a drug form. Now how do you decide that? How do you decide that it has a kashayatva, or how do you decide that it has a bheshajatva? Again, the Chakrapani has described it very nicely, my friends. Each and every person from Shastra, Dravya Gun, Bheshaj Kalpana, he should read this commentary very carefully. Every time I read it, I find some new things. Very important. Quality is essential to act as a kashai yoni, source of drug. What can be a source of drug? Drug material. Madhuradishu swarasaklakadi lakshana kalpana sambhavati na lavane. That I already explained you. Lavan rasa cannot produce less kalpana. Shakti vishesh kalpanartham cha kalpana kriyat. It should generate Shakti Vishesha. If it is not generating Shakti Vishesha, there is no point in producing any Kalpana. Shrita Shrita Fanta Kashayastu Dravyasa Kasnena Anupayojyasya Tattat Samskara Vasha Draveshu Dravyasa Stoka Vyavarupayeshartham Upadishyante. Very important. Dravya, any plant material or anything material, as a whole is not used as a drug. Many of that parts of that material are not useful to be used as a drug. They are waste product as far as drug is concerned. They don't have drug potential, or at least whatever drug you are planning, they have to be thrown out. For example, you are doing Swaras, you are taking out Swaras and the other material, you are throwing it out. So that is very important. Same thing regarding car. So पूरा के पूरा द्रव्य कभी भी ड्रग करके यूज नहीं होता है. कार्सने मीन पूरा, complete, as a whole drug, पूरा पंचांग or whatever you are using that word, that is not useful. Even food also, you grind the grains and throw out the whatever the husk is remaining. So that is important. So karsne, so there are three things. First is kalpana should be possible. Second thing, there should be Shakti Vishesha demand. Third thing, काrsne न अनुपयोज्य होना चाहिए. पूरा पूरा है तो यूज नहीं है. लवणे चैतन्य संबंधी, लवण के बारे में ये नहीं होता है. लवणम ही द्रव संबंधे सर्वात्मना एव द्रवम अनुगतम भवति. काrsne न अनुपयोज्य सही क्या नहीं, पूरा का पूरा लवण यूजफुल है. तस्मात् लवणम पृथक प्रयोगात् भात कल्पना असंभव चाचारेण कषाय संज्ञा प्रणयने रस्तमस्ति न निष्प्रयोजनेन आचार्य प्रवृत्ति. Remember very well. So that's why it is not possible to prepare a Kashaya Kalpana from Lavana potential, because it doesn't have potential to produce various drug dosage forms. It cannot produce additional therapeutic benefit, Shakti Vishesh. It cannot produce Shakti Vishesha. Dose form of formulation prepared when not all the matrix of the source material in reference is useful as a drug. Part of the material is only useful, not all the material. Dravyasa stoka means partly useful, not all. So dravyasa sokavya anupurushartham upadishyante. That is not possible with lavana. That is why it's not. Another very important thing, you are not using the whole drug, whole material. You are using its only active principle. The concept of active principle is also explained very nicely by Chakrabarti when he is explaining how the drug is to be used. What kalpana, what dosage form should be used. Because all dosage form cannot be used everywhere. It says Surasadinam Yathapuram Baradhikyam Phanta Chito Guru, etc. etc. Why? Because Dravya Sara Bhaga Mayatvena Attartham Guru Bahuve Dravya Karya Karaha. If you want maximum effect, maximum action, you should have maximum Dravya Sara Bhaga also. Dravya Sara Bhaga Mayatva, most important. Dravya Sara active principle, it should come. Then it becomes Guru, it becomes very heavy, it becomes very potent. And in that case, where you can use it, you can use only where the patient is strong enough or where the disease is very serious. Na sarvane surasadini sarvatra purushe jnani bhavanti. Very important. Kechit surasavya, kechit surapya, etc. I will not be reading it. So balavati purushe vyadho cha suraso yujjate. Why? Because dravya sara bhaga mayatvena. Dravya sara bhag mein hai. Na ayam malpavle purushe roge va yogvan bhoti bala bharuch veshiyate, etc. Untold effects will be produced if you are using the wrongly using a particular form. So that is why whenever you use a kashaya kalpana, vyadhi, atura, bala apekshani, important. It is vyadhi and atura bala apekshani. Apart from that, then a dravya apekshani tatha bodham. That means whenever you are preparing a drug form, you should have in your sight patient, disease and source material. It should be applicable. Accordingly, you have to prepare. Every kalpana is not applicable everywhere, my friends. Every drug is not applicable everywhere. Even the condition will be the same, no issue. But you have to consider this. That's why Even in the in next day, it's very important. See, whenever you are describing regarding the good drug, it said Bahuta tatra yogyatvam manake vidhakalpana. The good drug, which is the best material to prepare a drug. Bahuta tatra yogyatvam manake vidhakalpana sampacheti shukondarbhanam gunaichate. Sampat. I'm not talking about the other things, but I will be talking on yogyatva. Very important. It should be the material should be yogyatva, suitable to prepare a drug form. How do you decide? Tatra pratikartavye vyadho yogyatvam tatra yogyatvam. That means you have specific object in your mind. Your specific activity inside in your mind. Maybe that drug may be useful everywhere, but for your purpose, it may not be useful. People come to me whenever asking, sir, you have told that this is very good or that material is very good on the platform, and now I am coming to you and you are saying, no, it is not good for me because it's okay. Because general good and specific good is, you have to think it this way. You are a doctor. We are all doctors. We are physicians. We are Ayurvedic people. So we understand general good and specific good. Very important. When I go on this stage, I will say milk is very good for everybody. But whenever a prameha patient comes to me, I will say, don't take milk because it's not good for you. Because prameha hetu and milk hai bhai, paya. I don't know how many of you, the chikitsakas are advocating that don't drink milk to prameha patients. Prameha hetu mein hai. पहला तो है हमारा पर्पस कि निदान परिवर्जन और निदान परिवर्जन नहीं कर रहे हैं. So that because प्रतिकर्तव्य व्याधि हो उस व्याधि में इंपॉर्टेंट है क्या नहीं, क्या इंपॉर्टेंट है, that I will talk about. So that's why we are missing. Now that's why तदेव युक्तम भैषज्यम जद आरोग्याय कल्पते. Our main objective to give the health, promote the health. तद भेषजम युक्त युक्तम अलम रोग्यायति युक्त युक्त प्रवृत्तिमिति प्रवृत्तिश्च वैद्यानां चिकित्सा रूपा अलम समर्थम. It should be potential, should have potential to work as a drug in that specific condition. So you can't have any universal drug. You can't have any universal, what I would say, the immunity stimulant drug. Maybe for modern medicine, okay, because it's, if you are thinking Ayurveda, you can't have a universal immunity boosting or immunity stimulating drug in Ayurveda. I will explain it in the next slide. So it's very important. See what Charak says. It says, Roga bheshajayostu yojana yatha kartavya tad vaktum yojana chatushka. Yojana chatushka कहते हैं वो. क्यों कह रहे हैं वो? साच योजना रोगेण समम भेषजानां प्रायः शरीरमेव अपेक्षते. इते शरीर भेद प्रतिपादको अष्टौ निंदितो अभिधीयते. माय फ्रेंड्स अष्टौ निंदित अध्याय का जब वो डिलीट करते हैं तो वो कहते हैं व्हाई इज टेलिंग दिस अष्टौ निंदित, बिकॉज the ability of the body to accept that particular drug is different. I will give a selection of a drug in a disease always depends on the suitability of that drug in accordance with the physical status of that body. इसलिए वो बोल रहे हैं अष्टौनिंदित अध्याय. Physical state is important. I will give a simple example. Ashwagandha and Guruchi, both are very famous. People are using like tons of Ashwagandha and tons of Guruchi being sold as a very immunity stimulating, etc. And people are using it irrelevantly in every patient. Guruchi ले दो, Ashwagandha दे दो. अरे क्या कर रहे हैं आप लोग? आपने आयुष को दिया, सबको दिया, कितने लोगों के कॉम्प्लिकेशन हो गए? नहीं भाई, it's not applicable everywhere. What is, you have to see how it acts. Maybe the ultimate effect is its immunity boosting. No issue. We are not questioning that. But in which patient? In which person? Ashwagandha वृक्ष है, ब्रूहण है. गुड़े ब्रूहण है क्या? गुरुल is दीपक. गुड़वेल is given in persons who are debilitated. जो ये है क्या कहते हैं, जो obesity है, ये metabolic disorder है, गुड़वेल is good. But if you are giving ashwagandha is a bruhan drug in a very fat, obese patients, metabolic disorder patient, what will happen? It will increase okay immunity, but वो क्या होगा पता है? I know many cases of who are taking ashwagandha whose autoimmune diseases have been precipitated because of ashwagandha. It generates autoimmune diseases. I am telling you officially. I have evidences with me. I know a patient. I'm just now treating one patient who has had the autoimmune disease. Already he was having it increased by giving ashwagandha. Gout है, increase होता है gout. Metabolic disorder increase होती है ashwagandha से, नहीं दे सकते आप. है immunity booster, but and you are not doing that. What you are doing? You are taking one group of patients. वो कहते हैं immunity boosting, इसको भी दे दो, इसको भी दे दो, सारा सामान है. अरे नहीं चलेगा भाई. Why? What was happening? You are applying modern medicine principle to understand Ayurveda. Immunity boosting है ना, इसमें भी होगा, उसमें भी होगा, नहीं होगा, ऐसा नहीं होगा. You have to understand this thing. Most of the students, you are selecting certain drugs and trying to see the particular effect, antipyretic effect, hypoglycemic effect, and you are comparing that with some standard modern medicine drug. You are considering a hypoglycemic effect of a particular Ayurvedic drug with metformin. What? You are taking a joke, my friends. Metformin mechanism of action is totally different. Your drug is acting totally differently. Whether that particular action is required in that patient, are you going to see or not? You are not seeing dosha. You are not seeing dhatu. Nothing. You are not seeing agni. And what you do? After completion of the study, you collect the patient and then you divide them. Vataja, pittaja, kaphaja, etc., etc. Grave mistake you are doing. Just imagine that your drug is acting on only Vataja prameha, for example. And you are taking the random patients, and in those patients, you only have 2 or 3 patients of Vataja prameha, and all other patients are other dosha patients. Will be your drug active? No, it will not act. Because your base is wrong. Your principles are wrong. You have to consider all things prior to. You are not looking at that. I don't understand it. Many modern medicine persons, they are working on Ayurveda drugs. That's why they have established one traditional medicine research laboratory in Belgaum. They are working on that. You are also doing the same thing. Let them do it, but why? You are not for that, my friends. You consider all this and you prove that no, this is wrong. What are you doing? Shariram eva apekshat. Charak keh rahe hain. Sharir bhed pratipadako ashtav nindito ajiv bhedite roged. See, I am not telling anything by my own heart. I am telling what Charak was telling. If you are saying whatever telling me everything is wrong, then Charak was wrong. You accept that. I don't think Charak is wrong. We are not able to understand what Charak is going to say. Charak is saying. So very important. So unless you understand these things, you can't do research. You cannot practice Ayurveda. आयुर्वेद ड्रग यानी कोई नुस्खाबाजी नहीं है भाई. आप क्यों है समझते? नुस्खे बंद करो. नहीं है नुस्खे हमारे पास. कोई मुझे पूछता है कि भाई आपके पास डायबिटीज कम करने के लिए, मैं कहता हूं नहीं, मेरे पास नहीं है दवाई. हाँ, मैं डायबिटीज ट्रीट करता हूं, मैं दवाई मांगूंगा, नहीं दूंगा दवाई. ये हिम्मत रखिए. इट्स वेरी इंपॉर्टेंट फॉर यू पीपल टू अंडरस्टैंड. क्या कर रहे हैं आप लोग? See, even management of the disease condition, if you read Charaka, you can understand there is a sequence in management also. That sequence is also very important. You can see the first thing what they do, they tell you the food management. What type of food is to be taken, what should be prohibited, what are the abstinences, etc. You are managing the food management. Then they come to the therapeutic management. Then they come to therapeutic management. Again, they divide it. Santarpanjanya, apatarpana, depression therapy or repletion therapy. Whether it's bruhana or langhana, anabolic therapy, catabolic therapy. रसायन हेल्दी टिश्यू जनरेशन है या एक्सप्रेशन थेरेपी है, वाजीकरण वेरिटी ऑगमेंटेशन है या डाइजेशन है या पैसिफिकेशन है. Every drug is divided into these 6 therapeutic practices. Bruhana, langhana, rasayana, shodhana, vajikarana, pachana and shaman. Try to understand this and accordingly you take it up to understand. Now that's why you can see here the 6 Shatkriya, langhana, bruhana, rukshana, shrihana, swedana and shobhana. You have to understand the meaning activities also. Don't try to translate them haphazardly. Just to understand, whole understanding, they may not be a correct translation, but at least they are not misguiding you, this translation which I am giving. What langhana is doing is the catabolic therapy, which is depleting the mass, body mass. Bruhana is anabolic therapy, which is building up of the mass. Rukshana is desiccation, Snehan is hydration or lubrication, increasing the moisture in your body. Swedan is perspiration, whereas Stamban is anti-flow therapy. So this type of therapies, they are dividing it. Now, mechanism, I will come to most important part of as far as Dravya Shastra is concerned, is mechanism of action regarding the pharmacology. Because we are discussing the pharmaceutical science, that's why it is very important. People were till yesterday, people were shouting, "ये रस पंचक भंकस है, ये क्या बेरी देखते हैं, क्या द्रव्य देखते हैं, क्या रस देखते हैं, ऐसा कुछ होता है क्या?" Now modern medicine, they are understanding the significance of that things. Many people are coming out, my friends. Even the rasas, virya, they are understanding. Do you know? Recently, the Nobel Prize has gone to a person who has worked on. You might be knowing this thing. They have searched. They have researched on hot potency and cold potency. They have found out the receptors which are sensing hotness of a drug material or coldness of a drug. And that's why capsaicin was accepted as a drug which has been located by a receptor. I will explain in my later slides. And cold receptors are also there. That means to finding out how cold receptors and hot receptors, you did Nobel, and we're shouting about years times. Still, we have not proved it. They had done it. So that's why you know this shloka, Dravyam Gunena Pekana, this 5, 6, 5, Rasa Panchaka is there, Drakash. Now, how does it happen? I've been talking only on Rasa. Very important. Now, this Rasa, they have been described as ethnopharmacological descriptors. The term is new, recently coined, very apt term they have coined, ethnopharmacological receptors, descriptors. Now, see, test, most important, Rasa. Chemosensory cells are located in tongue. Up till now, I was speaking on Ayurveda. Now, let me come back to, come to modern medicine, where you can correlate whatever I was speaking up till now. Particularly the mechanism of action, Rasa Panchak. These chemosensory cells, they are located in the tongue. 5 basic tests modern medicine says: sweet, salty, umami, bitter, and sour. They don't accept pungent or astringent as test. They say that they are physical reactions, physical irritations. So sweet taste, salty taste, umami, bitter, and sour. Umami is, in Japanese, there is term umai, which means meaty. It is a meaty test, test of meat. And recently, another 6th test also they have identified. They say fat is also considered as the 6th test. Now, every test has its own receptors, test receptors. The test cells, there are 4 morphological subtypes. It may be a little bit repetition for you, but I am just laying down the foundation what I will be speaking in the next few slides. Type 1 is glial-like cells, detect only salty cells, salty test. Whereas type 2 cells, they are G protein converter cells, GPCRs. They detect sweet, bitter, and umami test. The sweet detecting receptors are T1R2, T1R3. They are not heterodimer, they are known as T1R2 and T1R3. Whereas bitter test is T2R. Umami is T1R1 and T1R3. You can see fat test, meaty test is classified in Ayurveda as a sweet test. And that is why you can see here also one test receptor, the dimer is common. The T1R3. T1R3 is common in both these things because they belong to one family, T1R family. Family is same. Sweet is T1R2, T1R3 heterodimer, whereas umami is T1R1 and T1R3. Whereas bitter test receptors are a different family. It is known as T2R family. Both are G protein coupled receptors, GPCRs. Very important. Type 3 cells are source stimuli. They sense the source stimuli out of one receptor located on the tongue. And the type 4 receptors are likely cells are progenitor because they generate cells known as progenitor cells. So these are the 4 morphological types of test receptor with tongue itself. Now, up till now, people were saying that tongue receptors are located only on tongue, but now they are accepting that receptors are distributed in the body. receptors are located throughout the body. Every cell our body understands taste. They have taste receptor. Particularly chemosensitive cells in the GI tract. That's right. A whole GI tract is distributed with these cells. Sweet taste receptor expressed throughout the body. Nasal epithelium, respiratory system, pancreatic islet, sperms, even sperms also. Testes, they have taste receptors. In enteroendocrine EEC cells, they are known as enteroendocrine cells. It is known as the enteroendocrine cells are known to be collectively largest endocrine organ inside the body. Try to understand the biochemistry. Don't go behind pharmacology. It is better go for food technology, food science, and biochemistry. Every Ayurvedic person should understand the food science and biochemistry. Forget about the pharmacology, my friends. I'm not saying pharmacology is not important, but our base is biochemistry and food science. Very important. Over 20 EEC types are identified. They secrete regulatory peptides and bioactive molecules, act locally on immune cells and neuroendocrines. See, this is the distribution which I am giving, just a table. You can see here different types of cells, A, and I cells, K cells, L cell, and the principal effects. Extra receptors, what they do? They control the appetite. They are the growth hormone release, facilitate intestinal motility. They trigger the emesis and nausea. They activate the gallbladder. They stimulate the insulin release. Very important. Stimulate appetite regulation, etc. Let us see some of the activities. See, you can see here distributed all over body. You can see here right from brain to foot, bone, brain to bone. You can see here. You can see bladder, pancreas, colon, stomach, sex, oral and extraoral places of expression of T1R2 and T1R3 alpha-gastrodestin receptors. Very important, my friends. Try to understand. Don't forget this figure. Functions of testosterone. I will be giving you functions of each and every testosterone very, very briefly. Of course, it's a very large one. I mean, it will take some time, but it's very important to understand you because this is the basic foundation lecture, and that's why if you move ahead with this understanding, I think many things will open to you. What are the functions of sweet taste receptors in GI tract? I'm not talking about taste located on the tongue. They are nutrient sensing. They initiate the glucose homeostasis. They secrete GI peptides. Intestinal mucosa is highly expressed with taste receptor proteins, including T1R2 and T1R3. Sweet taste receptor function as gut lumen nutrient sensors, help to regulate glucose balance and nutrient intake. Very important. Glucose balance they intake. Very important. Even insulin also, insulin intake also. In erythrocytes and neurons, I was talking about the GIT receptor. Now, in erythrocytes of neurons, what they do? These are glucose dependent. Role of sweet receptors in these things: tissue renewal from stem cells and precursors. Activation of the adipogenesis. Adipogenesis is the action of these taste receptors from pre-adipocytes to adipocytes. Very important. That is why in obese patient you should not give sweet, because the adipogenesis, it will enhance the stimulate the adipogenesis. They will convert the pre-adipocytes into adipocytes. That's why sweet is prohibited in obese patients. Sperm development and maturation, very important, my friends. That is why in infertility we promote sweet taste, dependent on transduction of sweet taste. T1R3 and G alpha, onset of spermatogenesis, rhythm of spermatogenesis cycle and aging of testes, very important. T1R3 inhibitors, if they are inhibitor, what will happen? Bitter test inhibits it. What happens? Lower the sperm count. That is why bitter test is contraindicated in patients who are having infertility. They activate the enhance the male infertility. Test inhibitor hai. Bitter test, very important. I'm not talking about any specific drug, my friends. I'm talking about test, because they are acting on this test receptor. That's why it's acting as a inhibition of this. Bitter test receptors now. It is a super family of 7 transmembrane G protein coupled receptors. Very important. T2Rs located in taste buds. Taste perception widely expressed throughout the body. Perception throughout the body. Extraordinary are attractive targets for targets of more than 50% of the drugs. My friends, most of the drugs, they are how they are? They are targets. The bitter taste receptors are their targets. That's why it is say it's a power. Good medicine always tests bitter. Why? Because they activate the bitter taste receptor. That's why 50. Even you take modern medicine or Most of the drugs are bitter tasting because they act on the bitter taste receptors. Okay. Now, bitter taste in the stomach, what they do? They confer protection against ingested toxin. That's why tiktarasa is said to be vishagna, because they prohibit the absorption of ingested toxins. Activation of bitter taste receptors in gut induces a release of ghrelin, an appetite stimulating hormone resulting in short-term food intake, deepen and passion. As soon as you take bitter taste, that's why initially if you take soup, bitter tasting soups you take, a vegetable soup you are taking because they are bitter tasting. They stimulate your appetite, but not too much because it's short-term there. It's a short-term food intake. Later on they will stop the food intake because they secretly release the release of ghrelin. This short-term food intake immediately followed by prolonged decrease in food sensation, delay in emptying time of stomach and leading to sensation of satiety. Very important. Non-lingual T2Rs, they are acting by 3 different ways of actions out there, location-wise. Cell autonomous regulation, paracrine regulation, and endocrine regulation. There are 3 methods because of actions of bitter taste receptors action. They have diverse function in different cell types or tissues. How they act? Shortly I will give you. Cell autonomous regulation, T2Rs, they are bitter compounds. What they do? In the motile cilia of human ear epithelia, elicit a dose-dependent increase in calcium ions, augment ciliary beat frequency. Very important. That's located in airway smooth muscles. What they do? Dose-dependent relax the pre-contracted airway. They dilate the airway. That is why Guruji was very effective, found it very effective in bronchial spasm in corona. Because it increased it dilated the airways. How? Ciliary beat frequency was increased. That is the action of the bitter, any bitter testing material. Even Kiratik, very important. Sudarshana, bitter testing. Gudvel, you take anything. They are acting like that. Because they stimulate the bitter taste receptors located over there, and they do that. Another important, paracrine role of the second mechanism of action. Reported in specialized small intestine into EECs. How they act? Increased calcium ion from T2R activation leads to release of cholecystokinin CCK through CCK2. Pumps bitter tasting toxins out of the cells. Toxins are excreted or through CCK1 receptor transmits signals to the brain to control the food intake. How do they act? Antitoxic effect. How do they do? By one way they excrete, they pump out the toxins. They prohibit the intake also, food intake of toxins, and that's why bitter taste was initially developed in the plants to resist the entry of toxins inside the plant. That's the main function. That's why they act as an antitoxic. Solitary chemosensory cells, third mechanism. A type of airway epithelium cell harbors T2Rs and taste transduction components. They are cremigna. How do they act as cremigna? Bitter compounds initiate a neurogenic inflammation to block bacterial invasion. They they create a neurogenic inflammation on the surface of the epithelium. And they act as they prohibit the bacterial invasion inside the body and decrease breathing rate and close the vomeronasal organ, or induce neurogenic inflammation in the nasal cavity, thereby prohibit the entry of bacteria or viruses inside our body. So ECs, enteroendocrine cells, they are scattered along the epithelium layer of GI tract from stomach to rectum. Everywhere you can find these ECs. Bitter testes or extracts from bitter herbs cause GLP-1 secretion in the human EC line in C1 H716, modulate food intake and regulate insulin release. Very important. That's why in Prameha, these ticks are important. T2Rs is negatively regulate TSH dependent iodine influx in thyrocytes, decrease the secretion of thyroid hormone. That's why they are important in thyroid also. Bitter testins potentially potent bronchodilators that also produce airway relaxation and protect against air. Regulate vascular smooth muscle contractility, very important. And that's why they T2Rs and test signal elements in GI smooth muscles, bitters induce a contraction when applied at lower concentrations and may play an important role in regulating cardiac functions as well, because they are just they are regulate the contractility, cardiac muscle contractility is regulated. In reproduction, bitter test is very important. That's why I'm taking it so much long. T2Rs and test transaction cascade components detected in different stages of spermatogenesis. Very important. Bitter chemicals induce a rise in calcium ion in spermatids. Induced spiralities indicate a unique T2R profile. Very important, my friends. Depletion of T2R 105 results in smaller testes and is sufficient to lead to male infertility, my friends. Very important. In female reproductive system, T2R38 is expressed in amniotic epithelium, syncytiotrophoblasts and decidual cells in human placenta and in placental cell line. So that is why they generate the infertility in female as well as male. That's why beta testing drugs are prohibited in pregnancy as well as in infertility in male and female also. In urethra, what happens? It initiates a bladder contraction. In a vivo study showed that oral gavage of the beta component chloroquine suppressed overactive bladder symptoms in mice that possess partial bladder outlet obstruction. Very important. Bladder contraction, they initiate the contraction. Weight reduction, most important. Metabolic syndrome. It is there is study. It resulted in gastro alpha-galactosidine weight loss with reduction in food intake. Effect of T2R decreased differentiation of pre-adipocytes to adipocytes. Sweet test was doing exactly opposite. Sweet test was stimulating pre-adipocytes to generate the adipocytes. Here beta test is exactly acting opposite. It is inhibiting the pre-adipocytes to generate the adipocytes. It is an antioxidant effect that I have told you because these chemicals delay the gastric emptying via ghrelin, decrease food intake, induce food secretion in colon, delay digestion and absorption of poisons from the spontaneous, flush out poisons from gut. Antitoxin. Now let us come to the salty test. Up till now we have talked about two important tests which are very important whenever you are treating a patient or even management of health also, bitter and sweet. Now the other tests, they are also equally important. Salty test perception again is a function of ENaC, epithelial sodium channel. They have appetitive response to NaCl, low salt linked to amiloride sensitive ENaC channels. Very important. 4 ENaCs have been identified. The only thing which I will tell, because it is taking too much time, just remember that they are working as an appetizer if taken in small amounts. Large amounts, it will prohibit that. Pungency perception, COTU test, transient receptor potential, TRP. What is the hot pungency? TRP vanillin type and TRP ankyrin type 1. My friends, the recent Nobel Prize is given for this particular invention. TRP vanillin type, TRP V1, V3 and V4, whereas TRP ankyrin type, TRP VA1, TRP1. And cold pungency is related to TRPM8. That means cold, that Shitavirya is sensed by TRPM8 receptor, whereas cold, the Ushnavirya is sensed by vanillin and ankyrin type of receptors. This is the recent development. Pungent compounds. This TRPA1 agonists improve intestinal transit. Very important. How do they do that? Example is 6-gingerol. It is an agonist acted on the capsaicin sensitive cholinergic neurons, moderated the contraction in isolated guinea pig ileum. This is the invention, current invention. They have worked on capsaicin. The Nobel Prize is given for this particular invention. In lower concentration, enhanced capsaicin induced contraction, while higher concentration inhibited them. They increase the contraction, intestinal contraction. Capsaicin stimulated motility of gastric antrum, duodenum, proximal colon when intragastrically administered in conscious dogs. Very important, my friends. Pungent. Vasodilation, pungent. Very important. Srotamsi vivronoti kotorasa. How does it do? Pungent agonist. Carvacrol, TRPV3 agonist. Vinol receptor. Eugenol from cloves. Induced TRP mediator vasodilation. They induce vasodilation. Heat and several mechanisms are involved. Decrease in the intracellular calcium ions of arterial mucocytes. Stimulation of nitric oxide, prostaglandin, intercellular derived factor, production of endothelial access. A number of actions are there. Every action explanation will require one full lecture on this one. But very important to understand, you can get everything on internet. You go there, don't simply go to internet to find out some reference. Go read full papers, very important. Kashayaras, again very important. Kashayaras, you know, most of the fruits they are having this kashayaras. Proanthocyanidins, a category of condensed tannins. Tannin is a very good anti-nutrient. You might be knowing, you might have, you will be listening to this particular word very first time. Anti-nutrients. Kudhanya varga, they contain anti-nutrients. That's why they are known as kudhanya. What they do, they alleviate diabetes and its complications, including hypoglycemic effect and decreased concentration of AGE products. Very important. Most of the times in metabolic syndrome, you find out that you have AGE products like acrylamide. AGE product is there. Tannin dressings. That's why we use tannin dressing, because they heal the wound. They have wound healing effect. Accelerate blood clotting. That's why kashaya stambak, we say ropak, stambak. Why? Because it heals the wound, accelerate blood clotting. Injection therapy with tannic acid produces a quick hemostatic effect in internal hemorrhoids. Kashaya is very important. Tannin decrease serum lipid levels. See, dietary prosanidins reduce triglyceridemia in vivo in one animal model. There are papers which are available. So kashaya is very important because of this proanthocyanidins and likewise other enzymes also. Valenoid challenge, Virya. I was talking about the recent lower price. This Valenoid challenges are related to heat sensation, involved in heat sensation such as that induced by chili peppers, capsaicin or ginger. TRP1 activated by pungent compounds, capsaicin, piperine, salty stimuli, low pH and non-oxidative. TRPM1 channels related to Shitavirya associated with cold sensations such as that induced by menthol. Nobel Prize for Physiology, David Julius and it has gone for this particular invention to find out the Ushnavirya and the Shitavirya. We were knowing that, but we were sleeping in our homes. Cold virya and trigeminal chemosensitivity, very important. Trigeminal nerve, oral mucosa contains numerous transient receptors, potential melanstine, like TRPM8 channels. These channels are receptive to cold stimulus. Phytochemicals introducing cooling sensation are TRPM8 agonists like menthol, geraniol, and eucalyptol. TRPM5 thermally sensitive. Its activity is increasing from 15 to 35 degrees centigrade. Therefore, responsible for the taste transduction triggered by cold stimulus. But the cold stimulus also stimulates the taste transduction. That's why large diffuse chemotherapy system, DCS, all over the body. It's an iceberg. Taste buds are most visible portion. This taste bud located on the tongue is an iceberg. Whereas extraoral taste are larger submerged part of these taste receptors. They are crucial for physical role, detection of irritant, control of airway surface liquid secretion, innate immunity, microbial population, regulation of appetite, cell proliferation, relaxation, contraction of muscles, bronchia, urinary bladder, vessels, and regulation of heart activity. Everywhere these taste receptors are acting, and that's why they are most important. So don't forget the Rasapancha. So my friends, with these words, I will take your leave. I tried to tell you what are the basic principles of Ayurveda. You should not forget those principles. Try to see how they are explained, what is the correlation, and I also explained you the significance of the Rasapancha in relation to the extra-gastric taste receptors. Thank you very much for giving me patient listening, and thank you, Mr. Organizer, for giving me an opportunity to speak on the first inaugural function. Thank you.
Thank you so much, sir. Sir have nicely explained about Ayurveda drug concept, mislinks, principles, and application of drug, mode of action, mechanism of action of the drug, application of scientific principles, and about research. Thank you so much, sir. Now session is open for audience. Anyone who have query can put your questions in chat box or unmute yourself, and you can directly discuss with respected sir.
Yes, is there any question? I will like to answer. No issue.
Please unmute yourself and ask your question.
Meeta ma'am raised hand. Please ma'am, unmute yourself.
मैंने नहीं किया था, वो गलती से चला गया, सॉरी.
Okay. Okay.
Please don't hesitate. I will try to answer the questions for my knowledge. No issue.
In chat box, Dr. Ramnathan have put something. Government draft. Yeah.
So Ramnathan, you can ask your question.
Mr. Ramnathan, you can ask your question. Please unmute yourself. Anyone else?
Student, you can also ask a question. If you have any query, you can ask.
You can send the questions later on also. Your organizers have my email as well as the WhatsApp number also. Don't hesitate to ask. I will try to answer as well, because I'm not a research scientist. I'm a simple learner of Ayurveda. I don't consider myself any expert or a legend, even though I have invited me. So don't hesitate to ask me anything by anybody. Don't hesitate. I am not an expert, natural expert. I'm not a research scientist. I'm a simple teacher. So you can ask questions, no issue. Don't hesitate. Don't consider me as a very big legend and how can I ask you? Don't do that. It's open. Yes, Dr. Katia, my friend, is asking something.
Yes.
Katia. Professor Savrikar, it was very enlightening lecture indeed. And because I was looking to listen from somebody the relation of Rasa, Guna, Veer, Pak, but at least started from Rasa at least, and how it affects the physiology and ultimately the pharmacology. And your description of the receptors, while you are giving lecture, I was looking at I was searching some articles on the net on the relationship of taste with the health. And some of them are good, but I think your collection is beautiful. And you have at least tried to explain in a very nice way on how Rasa Panchak affects the health. But one thing which is missing here, I think sometime later you may consider to link it with the dravya also. The receptors are there, but what kind of compounds these dravya have, which are binding with the receptors? Because if dravya is not there, the receptors are of no use. So maybe sometime next time you can discuss, or you are a very good writer also of the books and articles, I know it. So therefore, the way you have explained this, it's a very beautiful, very scientific way. And I think these kind of lectures are going to excite the students of BAMS and MD Ayurveda who have just listened that Ras Guna Veer Vipak and Ras Rakta Mans Med Asti Majja Shukra. This is the sequence. But what are the enzymatic pathway? What are the biochemical pathway? What basic researches have been done on this? I think that is the need of the day. If we have not worked, somebody else might have worked. And the way you have explained today about rasa through receptors, maybe the organizers may find some other speakers also who may also try to cover. And with Professor Savrikar, today it was rasa, tomorrow it may be other factors also, rasguna veer vipak. So I think it's a very enlightening, very knowledgeable lecture. I really gained a lot from the lecture, sir. Thank you very much.
Your presence is lot of us. Thank you, sir. Anyone else? Dr. Giriraj.
हाँ सर, नमस्कार सर। मैं सर ये पूछना चाह रहा था कि अभी कुछ समय पहले अग्निकर्म पे एक सेमिनार हुआ था राष्ट्रीय आयुर्वेद संस्थान में। उसमें एक जो चिकित्सक थे वो अपनी रिसर्च के बारे में बता रहे थे, CKD वगैरह इसके बारे में। तो जब वो औषधियों का चयन कर रहे थे पाचन का, तो अलग अलग डिजीज में जो अलग अलग जगह लिखा हुआ है, जैसे संतत जोड़ के अंदर जो दीपन पाचन औषधियों का बता रखा है, वहां से उन्होंने वो ड्रग्स उठाई और पेशेंट को दी। इसी प्रकार से अलग अलग औषधियों के अंदर अलग अलग जोड़ों के अंदर जो दी गई औषधियां थी वो अलग अलग तरीके से यूज कर रहे थे। तो मैं वहां पे समझ नहीं पाया कि वो किस प्रकार से मतलब उसका एप्लीकेशन कर रहे थे उस सीकेडी के अंदर या उस डिजीज पर्टिकुलर डिजीज के अंदर अलग अलग स्थान से उठाई गई औषधियों का चयन कैसे किया गया, ये थोड़ा मैं समझना चाह रहा था सर आपसे।
Sir, you are mute. Sabrikar sir, you are mute.
Yeah, very good question. Actually, one drug having a action, particular action is there. I have explained you examples of ashwagandha and gurve. Both are immunity stimulating agents, but they have to be used in different situations. Just like that, usually, whenever you select a drug, basically, drugs are acting as far as Agni is concerned. You have to consider the Dhattvagni and Bhutagni. Basically, Dhattvagni. Now, which particular Dhattvagni is disrupted, that you have to understand. This you can do very nicely by examining the patient, by his manifested symptoms and signs. And accordingly, if suppose Rasa Agni is distorted, then you have to give drugs which are acting on Ras. Okay? If it is Mouse Agni is distributed, then you have to act like that. You might be knowing that in Charak Jwara Chikitsa, ज्वर जीर्ण जब हो जाता है उसमें धात्वंतर प्रवेश करते जाता है, एक एक धातु में अंतर प्रवेश होता है, और हर धातु में प्रवेश करने के बाद कौन सा द्रव्य वहां देना चाहिए उसका भी एक्सप्लेनेशन वहां पर उन्होंने किया है, एक एग्जाम्पल के तौर पर। तो वो देखना जरूरी है कि भाई कौन सा क्या डिफेक्टिव है, कौन सा अग्नि डिस्ट्रीब्यूटेड है। मैं आपको और एक हिंट देता हूं, Agni are just like receptor. The concept of Agni and concept of receptor is not different. It is the same one. धात्वग्नि और भूताग्नि आपको पता है, समझ लीजिए कि पृथ्वी अग्नि है। पृथ्वी अग्नि कौन से अग्नि द्रव्य पर काम करेगा? केवल पार्थिव द्रव्य पर ही काम करेगा, वो अन्य द्रव्य पर काम नहीं करेगा। जैसे कि एक स्पेसिफिक रिसेप्टर रहता है, वो रिसेप्टर उसके लिए जो द्रव्य है उतना ही एक्सेप्ट करेगा, बाकी को छोड़ देगा। Just like that Dhattvagni is doing, and even Dhattvagni also. Dhattvagni will accept only that material which is for it, meant for it. Otherwise, all the material will be thrown out. It will be passed ahead or it will be thrown away. Yes. Just like that, in every condition, you have to consider which particular dhatu is distributed. And accordingly, you have to take into consideration which particular dravya is to be used. This is one way. Of course, other factors are also there. Just I am giving you some example I have given.
Thank you, sir.
Okay.
Thank you, sir. Anyone else? If anyone have any other question, you can send through mail. Sir have shared mail ID with us. We will share with you. Thank you, sir. Now, Dr. Mahendra sir. Mahendra sir. Okay. Now I would like to invite Dr. Sanjay sir for vote of thanks.
Can I step on you?
Yeah.
Thank you, Dr. Lewis, for coordinating this webinar series. Good afternoon to all. As we move to the end of today's webinar, I, Dr. Sanjay Kumar, Associate Professor in Department of Shastra and Research, Kalpana, NIA, on behalf of our department and NIA, Jaipur, take this opportunity to propose vote of thanks to all of those who have directly and indirectly contributed for the initiating this weekly seminar series. Today is a very auspicious day. We salute our Supreme Lord Ganesha, who has a curved trunk and whose radiance is equal to the sun. May Lord Ganesh blesses all our endeavors and remove all obstacles from our path in context of organizing weekly lecture series, especially on the course of Jigyasa. I wholeheartedly thank all the dignitaries present here for sparing their valuable time for us and gracing this occasion. At the outset, I thank our chief guest and resource person, Professor S.S. Salikar sir. We are really delighted with your knowledge and gracious presence and your visionary thoughts. Thank you, sir. I would like to especially thank our Vice Chancellor, NIA Deemed University, Professor Sanjeev Sharma sir, for his unfaltering support and confidence in us. Is the backbone of our institution and guiding force for every new step forwards. Whenever we would like to organize such type of any events, sir is always supporting in this direction. I would take this opportunity to express my deep regards to our Pro-Vice Chancellor, Professor Meeta Kotecha ma'am, Dean UG, and the HOD of Department of Draguna for being a constant motivator and guiding force in all our endeavors to excel in the field of education. A special thanks to the organizing committee, especially Professor Anupam Srivastava, for taking the lead. He is a very encouraging and enthusiastic person. I thanks and appreciate all teaching and non-teaching staff, especially Dr. Suman Sharma, Dr. Anita Sharma, and all HOD, Dean, and the non-teaching staff for their unflinching support and coordination. Heartfelt thanks to our scholars for their active participation. I also express my gratitude to the IT team from the RAV and the NIA for their technical support. My deep sense of thanks and appreciation goes to all the participants who attend this webinar with great enthusiasm and make today's event a great success. Even today is a holiday and most of the scholars from the Department of Rasastr and Department of Draguna and Department of Agatantra are still in this committee hall. So I'm thanks to all of you. In the end, I thank all the known and unknown hands that contributed for the success of these events. I would like to thank Professor C.V. Jha sir and Dr. C. Katiyar sir and many more stalwarts of Ayurveda to join with us. Thank you all for being us today. We hope this journey of knowledge will benefit all of us. Thanks one and all. Thank you all. Over to Dr. Ritesh.
Thank you, sir. Now, with the kind permission of our respected Vice Chancellor, sir, can we close this scientific session? Sir, please.
Yes. I am thankful to Professor Savarikar sir. Really, sir, wonderful thanks to you, sir. And thanks to all my all seniors and legends of Ayurveda and dear students and participants. And thanks to Professor Meeta ji and Jha sahab, Katiyar sahab, thanks from NIA and Ministry of Finance. Thank you very much. You can be speak now.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, everybody. Thank you, sir. Thank you for my views. Thank you, Nikhil. Thank you.
Thank you for being here.